Good morning, everyone. My name is Zarah Bantuas, and I will be your reporter for the endocrine disruptors, mesenchymal stem cells, and obesity. So let's start with the contents of my report. The contents of my report consists of the facts about the endocrine disruptors, how they interfere with normal hormone function, examples of the endocrine disruptors, diethylstilbestrol or DES, comparison of regulation of Mullerian ducts, about the bisphenol A or BPA, mesenchymal stem cells or MSC, stem cell nature of mesenchymal cells, finally, the tissues that contain mesenchymal cells. So let's talk about the facts about the endocrine disruptors. Endocrine disruptors can cause tumor as well as developmental abnormalities. Another few facts about endocrine disruptors, they are exogenous chemicals, which means they come from outside the cell. Next is that they interfere with the normal functions of hormones, which means they can affect the changes in your body. Next is that their alterations can only be seen microscopically. If their major alterations can cause physiological changes. So they produce functional changes which are subtle but can be extremely important phenotypic alterations. The next one is their effects can often manifest later in adult life and may persist to generations after exposure to the disruptor. Next one, they are everywhere, which means even if you weren't exposed to this when you were a child, you might still get it because it's everywhere, and both low and high doses can cause disabilities. However, the former can express its effects later in life. So the next one is moderate doses may be more dangerous than high doses. So now we're going to talk about how the endocrine disruptors interfere in the normal hormone function. The first one is they can mimic the effect of a natural hormone. Next is they can act as antagonists and inhibit the binding of a hormone to its receptor or block the synthesis of a hormone. Next is that they can affect the synthesis, elimination, or transportation of a hormone in the body. Some endocrine disruptors can prime the organism to become more sensitive to hormones later in life. It was long thought that there were only a few teratogenic agents in the environment, and that the only ones affected by these teratogens are the fetuses of women who were exposed to high doses of these chemicals during pregnancy. These teratogenic agents are what we know as classic teratogens. With that in mind, we can safely say that endocrine disruptors are teratogens. So now there are numerous endocrine disruptors. However, there were only 6 that were enumerated in the module. So these are diethylstilbestrol, DES, bisphenol A, BPA, phthalates, tributyltin, atrazine, and compounds released by hydraulic fracturing. Proceeding to the next slide. So now we're going to talk about diethylstilbestrol or DES. First, I'm going to introduce you to the important genes in the development of the female reproductive tract before going into what diethylstilbestrol, what is its effect to the human body. So now, we're going to introduce you to the WANT genes and the HOXA genes. I'm going to introduce you to WANT7A protein, which is expressed by WANT7A gene. It is responsible for the maintenance of HOXA gene expression pattern, and it activates and maintains HOXA10 and HOXA11 gene. It also activates another WANT gene, which is the WANT5A gene. So what is WANT5A protein? It is the protein expressed by WANT5A gene, which is necessary for the cell proliferation, HOXA10 protein and HOXA11 protein. I'm going to tell you more about what the HOXA10 protein and what the HOXA11 proteins are. HOXA10 protein. It is the protein which is expressed by the HOXA10 gene that is responsible for the regulation of the endometrial development during the menstrual cycle, and it is also responsible for regulating the or establishing the conditions necessary for the human implantation. Next is the HOXA11 protein, which is expressed by the HOXA11 gene, and it is critical for the development and maintenance of uterosacral ligaments and deficiency in pelvic prolapse. So now we'll talk about a brief history of the diethylstilbestrol, or DES. Diethylstilbestrol, or DES, was once thought to ease pregnancy and to prevent miscarriages during 1947 to 1971. Its usage was continued to be prescribed despite a 1950s study stating that there is no known beneficial effect to pregnancy until it was completely banned during 1971 by the FDA. So it was banned after clear cell adenocarcinoma was discovered in the reproductive systems of women whose mothers took DES during pregnancy. Another reason for the banned usage of DES is that it interferes with the sexual and gonadal development by causing cell type changes in the female reproductive tract. In many cases, the uterotubal junctions are completely absent, causing low fertility, infertility, and a high risk for reproductive health problems. So here we can see different types of risks in the health of the reproductive tract or the reproductive health problems. Here is a cervical dysplasia, which is a precancerous growth, tissue hood of the cervix, an adenosis of cervix and vagina, or the abnormal tissue. So we can see here. Another is the morphology of the female reproductive tract is a very different from the normal reproductive tract of a female. One is the most obvious one is the T-shaped or the constricted uterus, which means there will be there's slightly no room for the child to grow properly, unlike in the normal female reproductive tract, which is V-shaped or kind of a V-shape, which means there's plenty of room. Here I will just assume that this is where the absence of the uterotubal junction is. Okay, so here, and here is a very narrow passage, or a very narrow fallopian tube. So now, why low fertility and not complete infertility? If you guessed ectopic pregnancy, then you are absolutely right, because it's already seen here. So, what is ectopic pregnancy? This happens when a fertilized egg gets stuck on its way to the uterus, and often because the fallopian tube is damaged by inflammation or is mishapen. It also occurs when there is a hormonal imbalance or abnormal development of the fertilized egg also might play a role. Also, we've been talking about females. What about the male offsprings of the women who took DES? So I have this, this is just a trivia, quick one. So what happens to them is that male offsprings of women who took DES during pregnancy have high risk in testicular cancer and prostate cancer. So, the mechanisms of DES have been exposed after continued studies in mice were observed. DES syndrome, as it was said in the module, DES syndrome is the result of an altered HOXA10 gene expression. Normally, HOXA genes specify some regions of the FRT which are expressed in a nested fashion in the Mullerian duct. And since DES syndrome is the result of alterations or repression of the HOXA10 gene, the effect was especially seen in the stroma of the duct. So what is a HOXA10 gene? HOXA10 gene expression is necessary for the endometrial receptivity to blastocyst implantation. So the repression of this gene was strengthened by the phenotype HOXA10 knockout mouse, which then caused a transformation of the proximal quarter of the uterus into the oviduct tissue as well as the abnormalities of the uterotubal junction. So continuing, one link of the HOX gene and the uterine morphology is the WANT proteins. So, WANT proteins are associated with the cell proliferation and protection against apoptosis. Both HOX and WANT proteins are involved in the specification and morphogenesis of the reproductive tissues. HOX and WANT proteins communicate with each other to keep each other activated. However, DES resembles WANT7A knockout. And this resemblance, unfortunately, results in the repression of the WANT7A knockout, as DES acts through the estrogen receptor. So now we're going to have a comparison of the regulation of Mullerian ducts. A for the normal and B for the DES exposed. Here's a diagram of the normal function, and here is a DES daughter, or meaning DES affected or exposed. This repression prevents the maintenance of HOX gene expression pattern and also prevents the activation of another WANT gene, WANT5A gene, everyone, which encodes a protein necessary for cell proliferation. These factors specify and order the morphogenesis of the uterus. If proper activation of the HOX genes and WANT5A in the mesenchyme does not occur, it leads to a radically altered morphology of the female genitalia. So now let's talk about bisphenol A, or BPA. BPA, or bisphenol A, was first synthesized during the 1930s as an estrogen compound. It was found that it could be used in plastic production, and it is one of the most produced chemicals worldwide today. Human exposure comes primarily from BPA that leached from food containers, and this is because BPA does not always remain fixed in plastic forever, making it possible for anyone to ingest the chemical without knowing. Surprisingly though, the amount of the BPA is not that significantly large macroscopically in order for it to be biologically active. It only takes 300 micrograms per liter to cause chromosome anomalies. BPA is known to cause many things to not only the fetus but also to the mother. Supported by studies on mice and primates, BPA is known to cause ovarian and meiotic abnormalities, as well as aberrant follicle formation. It also causes BPA-exposed pregnant women to have high risk of miscarriages, cause abnormalities in fetal gonads, prostate enlargement, low sperm counts, and behavioral changes in later life when exposed in environmentally relevant concentrations. Bisphenol A also makes breast tissue to be more sensitive to estrogen, making them more susceptible to breast cancer in later life. It also causes early-stage cancer in mammalian glands in later life for fetuses exposed to the chemical. And it causes mice to become predisposed to developing cancer when they encounter estrogenic substances later in life. However, there are new studies that suggest that that may not be true for all strains of rats. So now we're going to talk about mesenchymal stem cells, or MSCs. Mesenchymal stem cells, or MSCs, are widely defined as a plastic adherent cell population. They can be directed to differentiate in vitro into cells of osteogenic, chondrogenic, adipogenic, myogenic, and other lineages. MSC renewal and differentiation potential are two criteria that define them as real stem cells. MSCs appear to lack one unique marker. In several studies, cultured MSCs have been characterized either by using cell surface antigens or by examining the cell's differentiation potential. In 2006, however, the Mesenchymal and Tissue Stem Cell Committee of the International Society for Cellular Therapy proposed minimal criteria by which one can define human multipotent mesenchymal stromal cells. So I will explain that mesenchymal stromal cells are actually cultured stem cells, basically. So, what are these criteria that make multipotent mesenchymal stromal cells multipotent mesenchymal stromal cells? These are that they must be plastic adherent state when maintained in standard cultured conditions and form colony forming units. Next, they must express CD105, CD73, and CD90, and lack the expression of CD45, CD34, CD14, CD11b, or CD19, and human leukocyte antigen D related surface molecules. What I just said was basically antigens and surface molecules. And another criteria or criterion is that they must differentiate into osteoblasts, adipocytes, and chondroblasts in vivo. So now we're talking about stem cell nature of mesenchymal cells. Stem cells are defined by their ability to self-renew and by their potential to undergo differentiation into functional cells under certain conditions. There is minimal evidence showing that such features exist in uncultured MSCs. However, cultured MSCs can self-renew and differentiate into osteoblasts, adipocytes, and chondroblasts in vitro. Whether these cells can also differentiate into tenogenic, myogenic, and numerous other cell types, as has been claimed in many studies, remains controversial. And we will know this in the next statement, which is there is no common progenitor for bone and muscle tissue after sclerotome myotome specification in somites, and hence it is unlikely that such a cell exists in the adult. Now, the supporters of this view claim that MSCs residing in the bone marrow can only develop into bone, cartilage, and adipose cells. And other researchers claim that MSCs can be isolated from the bone marrow as well as the other adult tissues and can be differentiated into epithelial, endothelial, hepatic, and neural cells. So, good day everyone. My name is Zarah Bantuas. I just noticed after editing my report that there were two unexplained parts on the report. Basically, that part, it was about the data found or collected that states that MSCs from extracted from different adult cells were they had different proliferative capabilities, self-renewal, differentiation potentials, and other capacities. And they're saying that they probably came from different types of cell origins, and they also probably have progenitor cells there, or progenitor mesenchymal stem cells. That's why they differ a lot in qualities that make them mesenchymal stem cells. So yeah, that's really just all I want to say. The findings of some studies suggest that multipotentiality of MSCs can be demonstrated by direct injection into extraskeletal tissues, such as the brain. However, studies have shown that injected MSCs were rejected or fused to existing differentiated cells within the tissue. The results of these studies further strengthen the elusiveness of finding a multipotential stem cell that may reside in all or most tissues. Nevertheless, this does not hinder the therapeutic potential of MSCs from bone marrow or other tissues. It may well be that MSCs, as they are currently used, are a heterogeneous population of cells, including both multipotent and progenitor cells with unique capabilities of tissue regeneration and immune response modulation. So, tissues that contain mesenchymal stem cells. The embryonic origin of MSCs is unclear. However, some findings indicate a possible origin of MSCs in a supporting layer of the dorsal aorta and the aorta gonad mesonephros region. It is consistent with the findings that MSCs or MSC-like cells were found circulating within human fetal blood. Another origin of mesenchymal stem cells are the bone marrow, along with the endothelial cells and the adipocytes. And these mesenchymal stem cells actually are the most well-studied mesenchymal stem cells. Mesenchymal stem cell population has also been identified in the bone marrow of the craniofacial complex. In adults, MSCs appear to be resident stem cells in many tissues, and they function in the normal turnover of these tissues. In adipose tissue, dermal tissue, the intervertebral disc, amniotic fluid, various dental tissues, human placenta, cord blood, temporomandibular disc, and peripheral blood. ASCs are similar to bone marrow derived MSCs, both morphologically and immunophenotypically. However, ASCs form more CFUFs when plated in culture, and also it is more attractive as a source of regenerative medicine or medical treatment purposes, because adipose tissues are relatively easier to obtain and can be collected with the use of local anesthesia. Also, there is only minimal discomfort and risk. Based on the current knowledge on mesenchymal stem cells, it is safe to state that they are associated with the microvasculature, and that different tissues contain MSC-like cells that have some characteristics in common and others that differ. However, they are all MSCs.